Well, welcome back. Today we have a very interesting topic, that is the second part what we have seen in the last video, that is chromosomal disorder. And today's topic is this, that is Mendelian disorders. Okay, now why they are called Mendelian disorder? Because they follow the Mendelian what you call pattern of inheritance. Well, one come from one parent, another come from other parent, then it goes to F1 progeny, then homozygous condition, dominant gene, a recessive gene, then in heterozygous condition, selfing, next what you call generation, then expression of recessive, and then what you call dominant. All this what we have seen in Mendelian inheritance back in that monohybrid cross, dihybrid cross, that pattern of inheritance is applicable to these genetic disorders. Because of that, these are called as Mendelian disorders. So the first point is clear why they are called Mendelian disorders. See, they occurs via single gene mutation. We have seen that change in single gene can lead to a mutation. Now, how that single gene mutation occurs, we'll see that in the latter part with a specific example of sickle cell anemia. Okay, and see, this is caused by single gene mutation. It follows Mendelian inheritance and it can be traced, it can be assessed, it can be diagnosed simply by pedigree analysis of the family too. That is the speciality of these disorders to remember 3 point: single gene mutations, follows Mendelian inheritance, and can be assessed by this pedigree analysis. And to go ahead, we should remember some of the names which is being given in NCERT as the famous Mendelian disorders. Okay, first here is Hemophilia, the royal disease that is being assessed in the pedigree analysis of the Queen Victoria of England. Okay, sickle cell anemia, which we are going to talk. Then phenylketunaia, will start the topic with that. Cystic fibrosis, color blindness, thalassemia. Okay, so what are the names? Hemophilia, sickle cell anemia, phenylketunaia, cystic fibrosis, color blindness, and thalassemia. So please remember the name correctly. These are following the Mendelian pattern of inheritance. Okay, and these are also genetical disease occurred by single gene mutation. Come to the specific topic as per NCERT. First one is PKU will take today. Okay, phenylketunaia. In one sentence, how to remember? This is an inborn metabolic error. See, it is inborn means it is inherited. And what is the error? The error or the mistake or the default is in metabolism. In our body, thousands to millions of reactions continuously occurs, and these are being metabolized by the biocatalyst, that is the enzymes, you guess. Okay, and these enzyme somewhere if not functioning, then there occurs the lack of metabolic functioning. That is called as metabolic disorder. Okay, now here this PKU, phenylketonuria, a condition, a result due to the defected gene on, remember correctly, chromosome number 12. Okay, so first thing, that phenylketonuria is a metabolic disorder because of non-functioning of enzyme. Where is the location of this mutation, mutant gene? On chromosome number 12. And which is the gene which is being mutated? See, mutation means mutare, to change, which is change, which is not normal. The gene that code for the enzyme phenylalanine hydroxylase. Okay, so remember the enzyme phenylalanine hydroxylase is the enzyme. Which gene, that gene that code for this enzyme is mutated. Okay, and that's why it is not producing that enzyme and resulting into metabolic error. Mechanism, how does it occur? See, phenylalanine is an essential amino acid. That means essential amino acid means the amino acid which is among the 20 amino acid, but this amino acid is not produced by body. So this amino acid is being brought from outside in the form of food, and then our body metabolizes that. That means it is being taken from outside and then it is being metabolized for further essential synthesis in the body. So these phenylalanine, an essential amino acid, when taken from food inside the body, this phenylalanine is being metabolized by phenylalanine hydroxylase, the enzyme. What does it converts? This phenylalanine during metabolism get converted to tyrosine, and we know that tyrosine is the one which is required for proper mental development, since it codes for what? The neurotransmitters you might have been guessed from class 10, dopamine or even norepinephrine. When phenylalanine metabolized by phenylalanine hydroxylase to tyrosine, that tyrosine converted into dopamine and norepinephrine. What happens? Due to mutation, due to lack of this phenylalanine hydroxylase, this phenylalanine is not metabolized, and it is being not converted to tyrosine, and hence there is no production of dopamine and norepinephrine. So what happens? This phenylalanine, due to lack of this enzyme, phenylalanine hydroxylase get converted to phenylalanine pyruvic acid, because it is not converted to tyrosine, and this phenylalanine pyruvic acid get accumulated inside the body. Now where is that deadly nature? It has all that ability to cross the blood and brain barrier. That means it can reach to the brain also. Now once it crosses the blood brain barrier, that means when it can pass inside the brain, in our brain there is one fluid, I hope you know that CSF, that is cerebrospinal fluid. How important the functioning of that cerebrospinal fluid is. It can get accumulated there also and inside the body also, and because of that its accumulation, it causes tremendous loss to the mental development and result in mental retardation. So we got phenylketunaia due to mutation. The gene for mutation, chromosome number 12. The enzyme which is mutated, that is phenylalanine hydroxylase. And what is doing? Because of lack of this enzyme, metabolic functions are halted for phenylalanine, and it is being not converted to tyrosine, which is not further converted to dopamine and epinephrine. And then what happens? It is being accumulated in the body as phenylalanine pyruvic acid. To test this, even if the testing is possible just 24 hours after the birth of the child, and this is very special. Here the blood sample is taken from the heel of that child, the heel stick of that child, and what is being tested? The ratio of phenylalanine to tyrosine. If the ratio of this phenylalanine to tyrosine is not what you call standard, then it is being diagnosed as the person, that boy is suffering from a genetic disorder, phenylketunaia. Now to cope on that, if it is a genetical disorder, very difficult to cure because it is from the birth, it is inborn. So the treatment is, of course, prevention is better than cure. We are taking phenylalanine from outside source. If we will not take phenylalanine inside the body, there is no need of its metabolism. To cope on that, to avoid the accumulation of phenylalanine pyruvic acid, better do not take the phenylalanine. That is the one precaution. Avoid the food with phenylalanine. Avoid proteinous food, since it consists of different amino acid. Proteins are made up of amino acids. Okay, so this is the treatment to avoid them. Standard certain enzymes are also available, which is being injected daily basis inside the body to cure this, but that is quite expensive. Move to the next royal disease, that is hemophilia. Now point wise we have to remember, first it is sex linked recessive disease, it is not dominant. Okay, it follows second point, crisscross inheritance. So first 2 points we will analyze. It is sex linked recessive disease which transferred by the X chromosome, that is the allosome. Okay, that is the sex chromosome. It is present in both X's in female and single X and Y in male. Okay, so it is X linked recessive disease. Why recessive? We'll see later. It follows crisscross pattern. Now crisscross pattern of inheritance means from father it will not go to the daughter. If it goes to the daughter, she will be only carrier. So if father is hemophilic, it will go to daughter only. Okay, I repeat, if father is hemophilic, this will go to only daughter. And when mother is hemophilic, it will come only to son. There are chances. How it is? What are the possibilities? Just understand. First, it is a sex-linked recessive disease. That means the mutation is present on where the X chromosome. It follows crisscross pattern, that is from father goes to the daughter, and from mother mostly it goes to the son. Okay, fine. Even if from mother it goes to the daughter, she will not be hemophilic, she will be carrier, because it is a recessive disease, must have to be present in homozygous condition. Okay, so we got first two things clear. It is a sex linked recessive disease. The chromosome is X chromosome on which it is mutated or present. Second fact, it follows crisscross pattern. There are a lot of possibilities, but in crisscross pattern, what to understand, a hemophilic father can transfer to the daughter, not to the son, never to the son, because from father only Y chromosome comes. Okay, then if it is passing to the daughter has to take one more hemophilic gene from mother, otherwise she will be only carrier. But if a mother is hemophilic or even a carrier, and if that X chromosome of mother goes to the son, then definitely that son will not be a carrier, because it will be hemophilic, since the boy composition is XY. If only a single X from mother hemophilic comes, he will not have another X chromosome to compensate the effect, lethal effect of this. In female, the condition is different. If suppose one X chromosome is normal and other is hemophilic, they will not suffer from hemophilia, because they have another X chromosome to compensate. So when the females are having only single X chromosome hemophilic, they are only carrier. But in case of male, if they are having a single, they basically have single X chromosome that is hemophilic coming from mother, remember, they will suffer from hemophilia, because they don't have another X chromosome to compensate, and which is present in females. Okay, now let us see one cross. We are just simply making a Mendelian Punnett square type of example. Hemophilic father. Okay, so we got directly its chromosomes as the gametes here. Okay, definitely the hemophilic gene will be present on the X chromosome. Y should be normal. We are crossing this with whom? To show the crisscross pattern, we are crossing this with normal female. Okay, normal female. See, take the gamete, one from mother, one from father. This will be a girl child, which should be carrier. One from mother, one from father. Now this is a boy and it is totally normal. This is carrier. Again, one from mother X chromosome, one from father. This is a girl child, but still she is carrier. Then take another possibility, X from mother and Y from father. Since it does not have that what you call hemophilic gene from mother, the boy is normal. What does it indicate? Simple, these two words. All the male should be normal, all the female will be carrier. When a hemophilic father marries with whom? A normal female. When a hemophilic father marries with a normal female, all the females' daughter will be carrier, all the males will be normal, since father has only one X chromosome and that is passed to the daughter, not to the boys. And since the Y chromosome is normal, so there will not be transfer from father to the son. Okay. Now, this disease, as I said, is also called as royal disease, because there is a lot of history beside that. That England's Queen Victoria was the carrier of this, and her entire family has destroyed a lot of what you call king's palaces in different countries. So that is a different story. We'll not go there. One of his son of just 30-year old just fall from the stairs, and because of that falling, started bleeding from his body, and just he died because of small bleeding. So that deadly is this, because once the person is suffering from hemophilia, even a small injury to the body can take out almost a heavy blood loss from the body, and that leads to the death. Okay, now let's move to the another topic, that is sickle cell anemia, to understand how that single gene mutation affects the personality. Okay, this was sex-linked recessive, this is autosomal recessive. Now autosomal recessive means it must be present in passing from the heterozygous parent to the offspring. Okay, if just one parent is heterozygous, another is normal, it will not be transferred. See how The gene for this sickle cell is in this 3 pattern. This is normal, or the hemoglobins are having intact amino acid as it is. So they are biconcave shaped, they are normal. This is heterozygous condition. Okay, one of the gene is mutated. This S stands for sickle cell. And here in homozygous condition, this shows the phenotype of sickle cell. So here the blood will be sickle shaped. Here the blood will be normal, but it is not completely normal. It will be carrier, since one gene is present to compensate, another is present as a carrier, but it will not express in single condition. It should be in homozygous condition. It will not be expressing in heterozygous condition. And see this condition here, both the genes are normal, so the person is normal. Here one gene is abnormal, mutated, the person is carrier. Here both the genes are sickle, the person shows the phenotype of sickle cell. How does this occur? A simple explanation is here. See, the hemoglobin chain has such sequence of amino acid. At first position it is valine, remember and listen carefully. Second position histidine. Third leucine, fourth threonine, fifth proline. Remember the 6 and 7 is glutamic acid. So the 6th amino acid is what? Glutamic acid. Now this glutamic acid has this codon CTC. Okay, so this codon CTC has anticodon from the tRNA during protein synthesis, that is C codes with gene, that is cytosine with guanine, thymine with adenine, and this cytosine with guanine. Okay, what happens due to single gene mutation in that codon, that is one nucleotide is now mutated, and because of that mutation, instead of CTC, what is being copied in the chromosome, autosome, that is CAC. C is normal, T changed, C is normal. Just single nucleotide changed. Now it is changed in the genetic makeup. Now when it is being copied in protein synthesis, what comes there? There comes the anticodon C, cytosine, guanine. Adenine, now there will come uracil. Cytosine again, guanine. What do you find? Instead of GAG, guanine, adenine, guanine, we have guanine, uracil, guanine. And that changes the amino acid, that is glutamic acid, is being replaced by what? The valine. So at 6th position in sickle cell blood or hemoglobin, at 6th amino acid position, instead of glutamic acid, due to single gene mutation, valine is being synthesized, and that changes the shape of this cell or RBCs from biconcave to sickle cell. So what is the logic? It is single gene mutation. Instead of glutamic acid, that amino acid is being replaced by a mutated mutare change amino acid valine, and because of that the mutant hemoglobin undergo polymerization at low oxygen concentration or a low oxygen tension, and that changes the shape of RBC from its normal biconcave to sickle cell. Okay, so what is sickle cell anemia? It is autosomal recessive. To transfer to the next child, at least both the parents should be heterozygous or suffering from it. And then what is its condition? In homozygous condition, it will express, show the sickle cell phenotype. In heterozygous, they will be carrier, and here it will be intactly normal. Which amino acid replaced? The 6th. What is that? Glutamic acid. Who replaces? Valine. It is a single gene mutation. So in this way, you can understand the pattern of this Mendelian disorder, its mechanism, why it is happening, and slight mechanism about what the treatment or extra matter is. So what is the goal for today? Remember these names of these Mendelian disorders, then the type, what you call the number of chromosome, and specific gene which is being mutated. In this case, how the pattern is, that sex-linked and recessive and crisscross pattern, and here in this autosomal recessive sickle cell, where is that mutation? It is in the 6th amino acid of hemoglobin protein, but glutamic acid is being replaced by valine. So it is, I think it is quite simple, and we'll have a very interesting topic ahead this, how this Mendelian pattern can be studied by pedigree analysis. Since these diseases can be traced by pedigree analysis, that we'll see in the next topic. So till then, just see this video once and twice and read in the NCERT. You will definitely understand it correctly. Okay. So thank you. Thanks for watching.